This is part 1 of 2 on chapter 3, Tools in Microbiology. This chapter is going to go through some of the techniques and tools that we're going to be using in lab. So we're going to be reviewing these throughout the semester in lab. In microbiology, we have something called the 6 I's. And it first starts off in the middle, and we actually have specimen collection that occurs first. And specimen collection, this can come from environmental samples. It can be specimens collected off of animals or people. So like urine, blood, water, just surface swabs, for example. After you collect the specimen, we're first going to go to the top right hand where it says inoculation. So usually after you collect your specimen, you inoculate some type of agar plate or some type of media, or it can even be like a live inoculation, so in a bird embryo, for example. So we need to inoculate some type of media. We then go down towards incubation. So we're going to go clockwise around the circle. So after you inoculate your sample, you have to incubate it. So this allows the sample to grow over time. And we'll be working with incubation quite a bit in lab. After incubation, you can isolate certain bacteria colonies and just focus on those species of bacteria. Most of the time in lab, we're going to be working with pure cultures, but every once in a while, I may mix cultures together, so you have 2 different bacteria, and then you can actually isolate the bacteria from each other. After isolation, you can inspect your bacteria, so this involves using microscopes. We also are going to be using lots of different dyes to help us inspect the bacteria, see what they look like. And during that inspection, you can gather information. So information gathering is our 5th I. So you can get this information from your inspection part. You can also do biochemical testing, which we'll do the second half of the semester. You can do drug sensitivity testing to figure out what drug works against your bacteria. You can even do DNA analysis to identify that specific species of bacteria. After you gather all that information, a lot of times you actually want to identify what the bacteria is, especially when we're looking at human or animals and we're helping to treat those humans and animals. A really important part of microbiology is learning how to use the microscope. And we're going to spend a whole lab on using the microscope and understanding, because you're going to be using it pretty much every single time in lab. So 2 really important things of a microscope, so 2 key characteristics of a reliable microscope are, 1, we need a microscope that can magnify, so we need to enlarge objects because bacteria are very, very small. So we need to make them bigger so we can actually see them. And then as we magnify our object that we're looking at, we also need to increase the resolving power, or the ability to show the detail in the image we're looking at. So this is showing magnification versus resolution. So magnification, you're just enlarging the image. So a really good example of this, if you think of like your digital camera, you can take a picture, you can zoom in, you can magnify the image, but a lot of times as you magnify an image, it gets blurrier. So that's where the resolution comes into play. So the resolution is just your ability to distinguish between 2 individual points, or to distinguish the detail. So resolution, this is analogous to pixels on a camera. So the more pixels you have, the higher resolution, the clearer the picture, especially when you zoom in on the picture and magnify it. So we need both of these things working at the same time in a really good microscope. The magnification part happens in 2 phases. So there's actually 2 different lenses the image passes through in order to be magnified. So one type of lens is called the objective lens. So the objective lens, we have 4 different objective lenses on our microscopes, and they're set at different magnifications. So the objective lens you can actually change or rotate. So this objective lens, it forms something called a real image. And then after the image passes through the objective lens, it goes through the second type of lens called the ocular lens. So that real image is projected into the ocular lens. These are the lenses you look through. In all of our microscopes, the ocular lens is 10x, so it magnifies 10 times. As the image passes through this ocular lens, it creates something called a virtual image. And this virtual image, it's inverted, so it's actually upside down and backwards to what is actually on the slide. And we'll be working with this in lab when we use the microscopes. So because magnification, it happens in 2 phases. To get your total magnification, you take your objective lens power. Remember, objective lens is the one that you can rotate or change. And you take this times your ocular lens, which again is usually 10. So objective lens times ocular lens, that gives you the total magnification that you're viewing the image at. You can also quantify the resolution, or actually give it a number. So your resolving power, it just equals the wavelength of light in nanometers, and this is usually given to you or you have to figure it out. And then you divide this by the numerical aperture of the objective lens plus the numerical aperture of the ocular lens. And your wavelengths can vary from anywhere from 400 to 750 nanometers. The numerical aperture, or the NA, it's written on the lenses, it's anywhere from 0.01 to 1.25. And we'll again be working in lab on this resolution and trying to understand what it actually means. So remember, resolution is the clarity of the image, or how well you can distinguish things when you're looking at them. For one of the objective lenses on your microscope, the biggest one, it's 100x, you're going to have to use oil in order to increase the resolution. So I'll show you how to actually do this in lab, how to use the oil, because you need to use the oil objective lens in order to actually see the structure of bacteria, and a lot of times to see the color of it. So basically what the oil does is it condenses the light, or it captures the light better than just what air can do. So it's going to increase the resolution. It allows more light to get through, so you get this clear image. This image again is showing resolution to help you kind of understand this idea. So at the top of the image we have bacteria cells that are considered to be non-resolvable. So you can't see them as individual objects. They're kind of smushed together. There's no space in between them. On the bottom we have the same image, but here our bacteria cells are very resolvable. You can actually see space in between your bacteria cells, so they're very, very clear. In lab, we're going to be using a compound microscope, and this is what it looks like. And I'm not going to go through all of the parts because you're going to learn about them in lab, but just to kind of familiarize yourself with the different parts, I can definitely show you. So we have the 2 objective lenses. So you have the 2 different types of lenses. You have the objective lens, and we're going to have 4 of them, including the oil immersion lens. So that's the first lens your image goes through. And then the ocular lens, those are way at the top. Those are what you look through. And again, our microscopes, the ocular lenses are always 10x, at least the ones that we have in lab. So different compound microscopes, or different optical microscopes, so these all use light in order to see the object. The microscopes we have in lab are considered to be bright field microscopes. So these are the most widely used microscopes, the ones you're going to see in labs pretty much everywhere. So in your image, when you look through these microscopes, your specimen is going to be darker than your surrounding field. So usually the surrounding field is going to be white or really bright. And we use these bright field microscopes for live and preserved stained specimens. And we'll get to different types of staining in a little bit. Other types of microscopes, you can have a dark field. This is basically just the opposite of a bright field. So your specimen is really brightly illuminated and the background is black or really dark. And these are used for live specimens, or they can be used for unstained specimens. Another type of light microscope is called the phase contrast, and this uses subtle changes in light waves that are passing through the specimen, and they turn it into different light intensities. So these phase contrast microscopes are really good at looking at the inside of your cell. And we have one microscope that does this phase contrast thing. And then we also have fluorescent microscopes that you can see. These ones, you use dyes on your specimen. These dyes emit visible light when we pass UV rays over them. So they fluoresce or they glow under UV light. And these are usually found in big research hospitals or research labs. They're really good at diagnosing infections. So we can actually see the bacteria. They're going to be glowing or fluorescing. The microscopes we've looked at so far give you a 2D image. If you want to get a 3D image, so you want to see the outside of the specimen, or you want to see through the specimen, you can use what's called a confocal microscope. So this uses a laser beam, so more concentrated light, and it scans the specimen. And it can actually scan the specimen at different layers, so you can have multiple depths or multiple planes, so you can actually go from the outside of your specimen and you can go through it using these scanning confocal microscopes. Another type of microscope besides the light microscopes are the electron microscopes. So instead of using light to form an image, electron microscopes, they use beams of electrons to form the image. So electrons are very, very tiny, if you remember from chapter 2, extremely tiny. These electron waves are 100,000 times shorter than the waves of visible light. So electron microscopes, they allow us to magnify and to resolve to very, very high numbers. So electron microscopes, they can magnify between 5,000 and 1,000,000 times. And our microscopes that we have in lab, they only magnify up to 1,000 times. So you can see that you can magnify a lot more with these electron microscopes. So light microscopes and electron microscopes, they pretty much have the same idea behind them. So you start off at the top. Light microscopes on the left, they start with light. Electron microscopes start with an electron gun that creates this electron beam. The light or the electrons, they first pass through a condenser lens, so you have to condense down the light or condense down your electrons. And then after that, they go through the specimen. That image of the specimen passes through the objective lens, so you magnify it the first time, and then your image will pass through your ocular lens, so you magnify it the second time, and you get to your image at the bottom. With light microscopes, we can view these images just with our eyes. With electron microscopes, the image goes onto a viewing screen. A lot of times nowadays it's just a computer screen that processes the image. And basically that's because you can't look at electrons, so you need to put it onto some type of viewing screen. There are two types of electron microscopes. So the first type is a transmission electron microscope, or it's abbreviated a TEM. Transmission electron microscopes, the electrons will go through the image. They're transmitted through the specimen. So electron microscopes, or the transmission electron microscopes, you get a 2D image. It's really useful for seeing what's inside of a cell. The other type of electron microscope is called a scanning electron microscope, or abbreviated SEM. Scanning electron microscopes, they scan the outside of the cell, so you get this really cool 3D image. And a lot of times they'll add artificial color to these 3D images, and they can be very, very cool and interesting to look at. And I'll try to incorporate some of these into lectures when we start getting into cells and looking at different types of organisms. So in our light microscopes, in lab we're going to be using light microscopes. We don't have any electron microscopes on campus. So for our light microscopes, or optical microscopes, we have to prepare our specimen in order to actually see it. So different types of preparation that we're going to be using in lab, you can use a wet mount. So this is where you just take your bacteria, mix it in with water, and you can look at it when it's still wet. Another type is called a hanging drop mount. A hanging drop mount, we can attempt to do this in lab. It's kind of hard to prepare. But those wet mounts and hanging drop mounts, they're used when you want to see bacteria alive and moving. So you want to keep them alive. The other type of mount that we're going to be using the most of is called a fixed mount or a smear, or it's called heat fixed in your lab book. So a heat fixed mount. So this, you start off with a wet mount. So you take bacteria, you swirl it into a drop of water on your slide. Except now we're going to let your slide dry, and then we're going to heat fix it. So you're going to pass it over a flame 2 or 3 times. And these fixed mounts or smears, the bacteria get fixed and they actually get killed on the slide. So when you look at them, they shouldn't be moving if you did it the right way. Once you have your heat fixed mount or your heat fixed slide, that's when you can start doing the fun part, which is the staining part. So staining, we use this to increase the contrast so we can actually see the bacteria cells are not just white blobs that you can't really see very well. So one type of staining, we can have positive staining. This is when your stain sticks to the surface of your microorganism. So your surface of the microorganism, they're usually negatively charged, and then you add a basic, or it's a positively charged dye. So your negative cell attracts the positive dye, and so your positive dye sticks to your actual cell. So positive staining, you're going to color your cell. Kind of the opposite of this is negative staining. So this is when your microorganism has a negative stain or negative charge to it. You add a negative or acidic dye to it. Negative repels negative, so you're actually staining the background, and your cells are going to appear white when you look at this slide. And we're going to be doing positive and negative staining in lab. Other ways to categorize staining, so some stains are simple stains. This is just where you use one dye or one stain to stain or give color to your bacteria cells. And this is usually done to reveal the shape, size, and arrangement of your bacteria that you're looking at. So I'm going to quickly run through the procedure of a simple stain. So if we start at the top left-hand side, you're going to spread your culture in a thin film over the slide. And usually you're going to spread it in water. You're going to let that water dry in the air, and then you're going to pass your slide through the flame to produce that heat-fixed part of the slide. Once you have your slide heat-fixed, you're going to flood the slide with a stain. So here we're using a pink stain. You're going to let that stain sit on there for the amount it says in your lab book, and then you're going to rinse the stain off with water and blot dry your slide. Then once your slide is dry, you can look at it on your microscope. And again, we're going to have to be using your oil objective lens, so I'm going to show you how to use the oil in the lab. Different types of simple stains you can use are, we have crystal violet, which is the image A on the right, it's the purple image. You have methylene blue, which causes the bacteria cells to appear blue. And then the third image, the pinkish red, that's a safranin stain. So we're going to be doing all 3 of these stains in lab to practice your simple staining. Once you have your simple stains done and you feel comfortable with those, the other type of stains we're going to do are called differential stains. These stains are a little more complicated. They have more steps to them. So you usually have a primary stain, and then in addition to your primary stain, you can have a counter stain. And this differential stain, it helps you distinguish between different cell types or different parts of your cell. And examples of a differential stain, you can have a gram stain, which is shown on the left here. And gram stain, we're going to do this multiple times. It's one of the most important stains that you can learn in microbiology. So in a gram stain, you're going to have your primary stain, which is crystal violet. It's going to cause the bacteria cells to appear blue or purplish. These blue or purple bacteria are considered to be gram positive. The counter stain is going to be safranin, which causes a pink or reddish to your gram-negative bacteria. Another type of differential stain we're going to be using is an acid-fast stain. These are good at identifying acid-fast bacteria, so Mycobacterium is an example of an acid-fast stain. So in the acid-fast stain, you have a pinkish primary stain, and then your counter stain is going to be methylene blue, and it causes anything that's not acid-fast to appear blue in color. So we'll be doing these, and again, these have multiple steps to them, so you're really going to have to pay attention. It takes a lot of practice to get a really good stain out of this. The third type of stain is called a structural stain. These stains, they reveal certain cell parts that are not revealed by just normal staining methods or conventional methods. So two examples of structural stains, we'll do a capsule stain. So some bacteria have a capsule around them, which protects the bacteria cell from dehydration and the immune system. So you can actually see the capsule. Another type of structural stain is a flagellar stain. This is where you can see the flagella, or the really long string-like structures coming out of the bacteria. And flagella, we'll get to this, but they're used for cell movement.